So now I would like to introduce tonight's speaker, Ryan Franke. He's one of our very own. He's a longtime MMS member, and he was the MMS newsletter editor from 2018 to 2020. He's also currently part of the mycology-focused forest pathology lab at the University of Minnesota. He researches biological control of invasive buckthorn and native fungi. If you didn't catch his last lecture on that last summer, you can find it on your on our YouTube channel, and we will pop that link up in the chat as well. If you're new to hunting, studying, or cultivating fungi, you may not know that ectomycorrhizal mushrooms are rarely cultivated because of the complex relationships that they have with other plants. It's usually the saprotrophs or rotters that we traditionally grow. So I'm excited for his presentation tonight, where he'll share successes and challenges to ectomycorrhizal mushroom cultivation. Thanks, Ryan, for bringing us this exciting content.
Yeah. Hey, everybody. Thanks for having me tonight. It's an honor to get to speak to you all and share what I've been learning about here at the University of Minnesota, where I've been studying for the past few years, primarily mycology and forest pathology. Okay. So I'll be sharing tonight the successes and challenges to ectomycorrhizal mushroom cultivation. Let's see, get my laser pointer out here. Um, if you've seen me present before, you probably know me for my work with using native fungi to control invasive plants like buckthorn. This line of research is more of a hobby for me, something that I picked up this past summer just because I was interested in the subject. And yeah, so I hope you all enjoy the presentation. So just first off, just a little bit about me before we dive into the subject matter. I became interested in trees at a very young age. Trees were, in fact, my first love, and fungi came second. It wasn't until a camping trip around Lake Superior during a chilly October that I first really noticed mushrooms in the woods, and thereafter became fascinated with them and started a hobby of cultivating mushrooms at home, which turned into a business. And then ultimately, I shuttered it after a few years to pursue some other opportunities. And during this time, I went back to school. I didn't have an undergraduate degree in hard science, but I went back to school for biology coursework at my local community college. And in 2022, I came to the University of Minnesota and got a job in the Borer Seabloom Lab of Ecology as a researcher. And during that time, I applied to the Department of Plant Pathology graduate program, and I got accepted and began working in Robert Blanchette's forest pathology lab on biocontrol of buckthorn using fungi. So there's me and my advisor, Bob Blanchette. Many of you probably know Bob. He's done a lot of work here at the U and is definitely a mentor of mine. And so I'm getting into the presentation for today. This is the outline of what we'll cover. First, we're going to talk about who exactly are ectomycorrhizal fungi, which I'll often refer to as EMF. Where are EMF found? And what do EMF do in their ecosystems where they're found? Second, I'll talk about why we care about cultivating EMF. What are some of the unique advantages of cultivating this guild of fungi? Third, I'll discuss some of the techniques and methods that have been developed for the cultivation of EMF. And then we'll jump into some EMF cultivation case studies, including chanterelles, as they've been researched in Sweden and Japan, the saffron milkcap, as it's been cultivated in New Zealand, and we'll bring it back home here to North America, looking at the pecan truffle and how it's been researched as a potential crop here in the United States. And then lastly, kind of just for fun, almost feels a little bit silly after these very serious cultivation case studies, but I tried my hand at cultivating um, some of these fungi this past summer. And so we'll talk about, and I'll just show you some photos from my spore inoculum experiment. Okay, so to start things off, I want to do a little activity. So this is interactive, and I want to do this because I wanted you to start thinking about um, how ectomycorrhizal fungi are not just the mushrooms that we know and love, like the morel mushroom or the chanterelle mushroom or the porcini mushroom, but they are also the underground vegetative part of the fungal body called the ectomycorrhiza. And so you can see here, there's two photos here of different species of EMF. And what's pictured is their ectomycorrhiza, which is essentially like a fungal sock over a root tip. And it is this structure that does the metabolism and is present in the ecosystem year round. And so these are really important structures for these fungi. And so linking the underground part of the fungus with the above ground part of the fungus, I'd like you to take a guess at, we have the pecan truffle at the bottom of the screen, which ectomycorrhiza on top produces the pecan truffle. So just go ahead and type it in the chat. And so if you guessed that the ectomycorrhiza on the left is the pecan truffle, you are correct. This is tuber lyonii, and you can see there is some similarity in the coloration of the ectomycorrhiza to the coloration of the fruit body. And so this one's kind of intuitive. And so here we have another one of these. We have the fly agaric mushroom. And so you can take a guess, put your guess in the chat, whether the ectomycorrhiza on the right or on the left belongs to the fly agaric mushroom. Okay, we got some for the right, a couple for the left. Okay, yeah, so it is the ectomycorrhiza on the right. So if you guess right, you're correct. Again, you can see some similarity in the coloration of the stipe and the gills of the fruit body, matching the coloration of the mycelium in the ectomycorrhiza. So yeah, this is Amanita muscaria. Another one of these, we have maybe a lesser known fungus. We have Piloderma olivaceum, and this is the fruiting body here at the bottom. And you can go ahead and take a guess which ectomycorrhiza, type it in the chat, produces this fruiting body. Heather says she's going to keep guessing right because eventually it will be right. Yeah, so everyone's saying right. Uh-huh. And again, yeah, so the coloration of the ectomycorrhiza matches the coloration of the fruiting body. So you all are correct who said right. There we have Piloderma olivaceum. And I think this is the last one of these. This one's a little bit more challenging. So go ahead and take a guess. We have the porcini mushroom. It's produced by one of these ectomycorrhizas. Okay, we got some folks saying, well, a lot of folks saying left. And you all are good. Yeah, so yeah, Boletus edulis is the fungus on the left there. And yeah, that one wasn't so intuitive for me, but you all just knocked it out of the park. So let's move on. So here they are, the ones we just saw, as well as some other new ones. We have slippery jacks up on the top left, letter A. We have Amanita muscaria, letter B. Boletus edulis, letter C. And then we have Astraeus territus, letter D. And so um, something that these ectomycorrhizas really have been, I guess, has been extremely difficult to challenge or to study them or to work with them in the past. I would say before the advent of DNA sequencing, because if you dig up a root tip in the woods, just because there's a fruiting body above that root tip, um, that doesn't necessarily mean that the fruiting body will match, because oftentimes the host can be colonized by multiple species of EMF at one time. And so oftentimes people would, dig up these root tips and they just they wouldn't know what it belonged to. But with the advent of DNA extraction and sequencing, the DNA can be extracted from these ectomycorrhizae, sequenced, and then identified to species. And so now scientists can go out and actually study the ectomycorrhizal community via root tip sampling, which has a lot of advantages in comparison to studying the community um, based on visual observations of fruit bodies, because the root tips are there 365 days a year. So as long as you can dig the soil, you can sample them. And also, some EMF may produce more fruiting bodies than others. And so it can give a skewed idea of the relative abundances of these fungi in the community. Um, Also, something that we'll talk about later in this presentation is that now that we can identify these fungi from DNA sequencing and we can know which species an ectomycorrhiza belongs to, we can actually isolate the fungus from the ectomycorrhizal root tip, which, as we'll see, has been demonstrably easier to do, orders of magnitude easier to do than isolating from the fruit body tissue. And so this kind of opens up our possibilities of working with these fungi.
Hey, Ryan, can I insert one question before you move on?
Yeah.
We had piece someone asked, are the root tips usually from trees or are they all from plants? I mean,
Yeah, that's a really good question. So, ectomycorrhizal fungi pretty much exclusively colonize trees. There's another, um, I would say, phylogenetic grouping of fungi called the arbuscular mycorrhizal fungi, and those fungi also associate with plant roots, but they primarily associate with non-woody plants. Um, so it's kind of an interesting division. However, some of those AMF species, the arbuscular mycorrhizal species, also associate with trees. But I don't know of any ectomycorrhizal fungi that associate with herbaceous plants. But there's probably an exception to the rule out there. It's biology. There's always exceptions in biology. But yeah, good question. So if we take a closer look at these ectomycorrhizal structures underneath a microscope, this is what you would see. Um, we have, this is an ectomycorrhizae on a beech root. So this is a big cross section of the root. And you can see down at the bottom here, these big cells are the root cells, the cortex cells in the root. So this is plant tissue. And then in between the plant tissue cells, we have fungal tissue. And this is referred to as the Hartig net portion of the ectomycorrhiza. And it is between the cells in the Hartig net that nutrient exchange happens between the fungus and the plant. That's basically what's going on here. Is it's a mutualistic symbiotic relationship between the fungus and the plant, and they cohabit together, and they both provide the other symbionts some kind of unique benefit that they wouldn't be able to obtain on their own. And so you have the Hartig net, and then out external to the Hartig net, you have the mantle. And this is the fungal sock that I was referring to earlier. And this is what we see when we look at an ectomycorrhizae with our naked eye. And you can see it's lots of cells thick, and it doesn't have the normal filamentous structure that fungi normally have when they're in their soil environment or even in the Hartig net. And then what you can start to make out here is some external to the mantle, some hyphae that goes out and explores the soil environment. And so that's the hyphae that will go out and form fruiting bodies above the above ground. And also it'll go out and explore the soil environment for nutrients and for water. Okay, so now that we have some basic understanding of what EMF are, I want to talk about who they are and where they are found in the world. EMF comprise about 8,000 to 55,000 species worldwide. These are estimates based on basically some equations that take a sample set and then extrapolate to what the total population of species could be based on a limited sample. And so that's why there's such a big range there, is that we haven't observed 8,000 or 55,000 EMF species, but we estimate that there probably are about that many species. And the EMF lifestyle of forming ectomycorrhizae has evolved more than 80 times independently. And this has happened basically where you have a humus decomposing fungus or a wood decomposing fungus, basically evolving these ectomycorrhizal structures and taking on this new lifestyle. And this has happened in the Ascomycota many times, the Basidiomycota many times, and to a much lesser extent, the Mucoromycota. And those are all phyla of fungi. Most of the mushrooms that we know and love and that we collect here in Minnesota that are EMF are probably in the Basidiomycota. There's more than 250 known genera of these fungi. So, this is a really successful evolutionary strategy. There's a lot of diversity, and that's part of what makes this group of fungi so interesting and exciting. Now, while only 2% of plant species will form symbiosis with EMF, 25% of global vegetation cover is associated with EMF. And so you could think about it how the partners, there's not a whole lot of plant partners for EMF, but the ones that do exist, primarily trees in the Pinaceae, Fagaceae, Betulaceae families, are very numerous, are very abundant on the earth. And so these EMF are also equally abundant on the earth and really important part of the ecosystems where they reside. And so this equates to 60% of global tree cover, totaling about 1.8 trillion trees that are associated with ectomycorrhizal fungi. And you can see from this figure here showing basically lighter colored areas, greener areas have more EMF species present. And you can see that EMF richness and diversity is really concentrated in temperate forests and boreal forests throughout the world. And this is interesting because this is I would say, an inverse trend of what we see with plant biodiversity, where we have the most biodiversity at the equator, and then you have less biodiversity as you move toward the poles. But again, this is because these fungi associate with these temperate and boreal species of trees, that they are located in temperate and boreal areas. And as I said earlier, one tree can host, dozens and dozens of species, or maybe even more than 100 species of ectomycorrhizal fungi. And so there's a lot of diversity. And let's see, here we are in Minnesota. If we were to zoom in, we could see basically we are in a biodiversity hotspot for EMF. The blue here is showing basically prairie. This is all kind of prairie, but where we have the eastern broadleaf forest, which Minnesota is right on the edge of that biome, the eastern broadleaf forest in Minnesota is very rich with EMF. And, hence we have a very strong mycological society here in Minnesota, I think partially for that reason. So what do EMF do exactly? Well, we touched on this a bit earlier, but ectomycorrhizal fungi are essentially associates of trees' roots. And what's going on here is that the tree is photosynthesizing and taking those photosynthates and shunting them down, sometimes as much as more than 50% of the photosynthates are shunted down to the roots and are shuttled into their fungal partners. And the fungus uses those sugars for their metabolism. Now, the fungus, on the other hand, accumulates, absorbs nutrients like nitrogen, phosphorus, potassium, micronutrients as well as water from the soil, and shuttles those nutrients and water to the plant roots. And as you can see here, the plant roots occupy a very small volume of the soil, and the mycelium increases their volume by exponentially. And so the plant roots now has access to a larger volume of soil, where it can benefit from gaining additional water and nutrients. And so this is a win-win. This is a mutualistic symbiosis. And so the ecological consequences of this symbiosis are many and complex. And just to name a few here, there's been lots of scientific studies that have shown enhanced plant host resistance to root pathogens when those hosts have an ectomycorrhizal fungal partner. Also, enhanced plant host resistance to abiotic stressors like drought. Also, heavy metal accumulation in the soil. These kinds of things can have a negative toll on plant fitness, but with an ectomycorrhizal fungal partner, the toll on the fitness is decreased. Also, these ectomycorrhizal fungi, are composed of sugars that originally came from the photosynthates that were manufactured from the CO2 in the air. And so this is kind of like you can imagine a pipeline for the CO2 moving from the air, the atmosphere, into the plant leaf, down into the plant stem, into the plant root, and now into the fungal body. And so carbon is being sequestered into the soil via this relationship. And Peter Kennedy's lab has done some neat work looking at fungal necromass in the soil. And so some of these ectomycorrhizal fungi have kind of highly melanized mycelium, and the mycelium is recalcitrant to decay. And so that carbon that's stored in their body is not just being decayed by bacteria or some other fungus and then respired back into the atmosphere, but it's actually staying in the soil for lengthy periods of time. Lastly, I want to touch on the wood wide web. And this is a concept that was first put forward by Suzanne Simard, I believe in the 90s, who was a forest ecologist. And she set up an experiment basically using tagged carbon dioxide and being able to trace that carbon dioxide, that isotope, through a plant's metabolism as it photosynthesized and fixed it into photosynthates, and then watch those isotopes of carbon move throughout the underground soil economy. And she did this in I believe, a Pacific Northwest forest where there was fir and there was birch. And there was some fir that were dominant, it was birch. I think it was birch that was dominant in the overstory and fir in the understory. And she saw that there was via what was assumed to be a mycelial network, the flow of carbon from the birch to the fir and also from the fir to the birch. However, the net flow of carbon occurred from the birch to the fir, and so from the dominant overstory tree to the seedling. And this is kind of an astounding phenomenon where you have a species of tree that is benefiting a different species, which goes against what we learn about in evolutionary biology, that species will do everything that they can to increase their own reproductive power and increase their ability to produce offspring. And so these mycelial networks appear to be coordinating the transfer of nutrients in forest ecosystems, because one network can occupy the tree roots of multiple individual trees at the same time. And this has been hypothesized to maintain biodiversity in forests and also stabilize forest ecosystems. So that way, when the birch trees all grow big and they start to die due to old age, that there's something underneath them to replace them, like fir trees. And so, yeah, that's a neat concept. Now, I just want to touch on arbuscular mycorrhizal fungi because you may have heard this word AMF before. And this is I don't want you to be kind of confused or wondering like, wait, what about AMF and how does that work with EMF? Well, as I touched on earlier, these are two very different lineages of fungi, and they also operate pretty differently within their plant hosts. Um, and so we have here a microscopic image of arbuscular mycorrhizal fungi associated with a plant host root. And basically, they not only grow between plant cells, as you can see happening here, like the EMF, but they will also grow into the plant cells. These kind of vague looking blobs here are called arbuscules. And those they actually invaginate the plasma membrane of the plant cell, and so they're within the cell wall. And it's within the cell that they do nutrient exchange with their plant hosts. And arbusculo means little tree in Latin. So these, if you had a clearer image of this, they kind of look like little branches within the plant cell. And also, they form these vesicles, these lipid storage organs within the plant host, which is unique to arbuscular mycorrhizal fungi. And these are also very important for ecological health and are quite significant on a global scale. Just as I would say, just as significant as EMF, but more so in tropical and subtropical ecosystems. Also, to be mentioned is that these fungi don't produce large, fleshy fruiting bodies. However, they do produce spores underground that are quite large, and you can see them with the naked eye. But we usually don't see them because they're being produced in the soil. And they're anywhere from 0.04 to 0.4 millimeters in diameter, and you can see a whole variety of these pearly looking spores, and they're just beautiful in their own right. Okay, so I think this is why a lot of us got into mycology in the first place, is that some fungi produce delicious and beautiful looking mushrooms. And so these are all species of ectomycorrhizal fungi that we have here in Minnesota that we know and love. And some of these have been under attempts for cultivation. And some of them have had some success, and others have been under attempts for cultivation and have had basically gone nowhere. Here we also have these truffles, which are in the tuber genus, and they're different from their basidiomycete counterparts, which are all those in the top row. Well, one, because these are ascomycetes, and so they're a very different lineage of fungi. But also, these fruiting bodies are produced below ground. And so the spores have different dispersal mechanisms. Namely, they have a very fragrant scent that attracts different insects and animals to come and chew on them and basically carry them throughout their subterranean environment and spread their spores to new plant hosts. And so we'll also talk about these fungi because they produce these mushrooms that are highly sought after. They're very fragrant and they're edible and delicious. And so yeah, we'll touch on those today too. So before we get into the techniques and some of the case studies for cultivating these fungi, I want to just call out some of what I think are the unique benefits of cultivating EMF mushrooms. And I think these are reasons why this research should get funded, and reasons why their cultivation kind of goes, I guess negates the idea that why not just go get these mushrooms out in the woods like we've done forever. And so let's start off here. Letter A, we have these kinds of agroforestry systems that include an ectomycorrhizal fungal component, offer both a carbon storage and a protein benefit at the same time, which is really unique. All other major land management systems that produce protein, like poultry or grazing cattle, all have a net carbon, what should I say? They're all carbon positive. So all the carbon flux is positive, so they're emitting carbon. However, an EMF agroforestry system is actually estimated to be net carbon negative. And so there's these researchers in Scotland that are interested in the subject, and they basically took some yield data from a New Zealand experimental orchard that produced about 1,100 kilograms of saffron milk caps per hectare, which is a lot, and estimated that assuming this yield, 406 kilograms of CO2 could be stored per year per kilogram of mushroom protein. And this is in contrast with poultry systems, which are estimated to emit between 10 to 30 kilograms of CO2 per year per kilogram of poultry protein. And so potentially we can, have our cake and eat it too. Now, second, an EMF agroforestry system could provide a secondary income source for farmers. And so because these fungi need a plant host, well, a lot of their plant hosts are commercially interesting tree species. And so we have these spruce or fir trees here that are in a Christmas tree plantation. And so some of these fungi could be double cropped with Christmas trees. We have a timber plantation here. These fungi could be double cropped with Scots pine or red pine or white pine. And here we have a hazelnut plantation, and hazelnuts can host, lots of different species of EMF, including many edible ones like the hedgehog mushroom and the chanterelle mushroom, and also tuber species, so truffles. And so, and hazelnuts are not the only nut crop that can host these EMF. Other nut crops include pecan and Korean nut pine. And so there's just a lot of potential here to stack functions and gain multiple benefits out of one agroforestry system. Now, third, I think, this just seems reasonable to me that EMF mushroom cultivation, given that techniques are heavily researched and optimized, that this could be more efficient than saprotrophic mushroom cultivation. It could be less labor intensive as well as use less plastic. And so, as I mentioned in my intro, I used to grow mushrooms for a business, and I grew lion's mane and shiitake and oyster, and I grew a lot on logs, but I also grew quite a bit in 5-pound sawdust blocks. And these sawdust blocks are basically inside of a plastic bag that is thermotolerant and goes through sterilization with steam. And then you have a sterile substrate that you can inoculate your fungus into, and it colonizes the substrate and then fruits from that sawdust block from within that bag. And this uses, a lot of plastic. This is my least favorite part of mushroom cultivation. And I think a lot of mushroom cultivators would tell you a similar thing, that they just fill up like dumpsters and dumpsters worth of these plastic bags each week. And however, in contrast, EMF fungi in an agroforestry system would not require plastic to house its fruiting substrate, because the fruiting substrate is simply the soil. So you could do away with these plastic bags at least in large quantities. And lastly, I want to mention that having, a larger supply of these fungi coming from cultivation could reduce foraging pressure in natural ecosystems. And I don't say this because I believe that picking mushrooms in the woods decreases future abundance of those mushrooms in the woods, because that's been disproven. But simply I say this because, there's just not enough really great chanterelle spots around the Twin Cities. And wouldn't it be great to go and be able to buy these things freely at the grocery store, just like you do white button mushrooms or shiitake? Okay, so now that we've reviewed some of the basics of EMF and why we should think about cultivating them, let's talk about some of the techniques that have been developed for their cultivation. Each EMF species will have kind of different requirements for growth. However, some of them can be actually grown pretty easily in culture, just like an oyster or shiitake mushroom. And so, in case you're unfamiliar, these are petri dishes here, and these petri dishes have an agar media in them, which is basically like a gelled, kind of like a jelly, like a firm jelly that has different nutrients in it. And oftentimes for saprotrophs, you put in some malt extract, just like you would use when you brew beer or something, and maybe a little bit of yeast extract, which you can also buy in the grocery store. And that's in some agar, and you autoclave it, and that's your media. You pour it in your petri dish, and it gels up, and you can grow your fungus on it. And that can work for a lot of, well, I wouldn't say a lot, but some of these EMF species too. However, others have more fastidious or picky requirements for growth and require the addition of different micronutrients, or I should say vitamins, like thiamine or biotin. And some of them just won't really grow at all. And unfortunately, this can be the case for some of our most beloved mushrooms. Like chanterelles are just notoriously difficult to grow in culture. However, this example here, we have Pisolithus, which is an ectomycorrhizal fungi that associates with eucalyptus plants. And so what these people, the authors of this paper did was they grew out their fungus on an agar plate, and then they germinated their seedling on the plate. And then they incubated the plate vertically so the seedling could grow up. And as the stem shoots up, we have the roots growing down, and then we have the fungus basically forming that ectomycorrhizal structure, the Hartig net and the mantle around those roots there. And this can take, up to 3 or 4 months or even longer for the ectomycorrhizal structure to be well developed. And you can see that happening down here too. This can also be scaled up into these jars, which I thought was a neat idea. You just have the agar media there, the fungus, which is orange, and the plant root shoots that are green. And then this is a cool photo. This is from Dr. Louis Mielke, who is a friend of mine, and he's from Minnesota. He went to the U of M, and then he did a PhD in Sweden, and now he's doing a postdoc at the University of Stirling in Scotland. And he's working on an afforestation project of the Scottish Highlands as like a carbon drawdown strategy. And so these Scottish Highlands are really harsh. And so what their project aims to do is to mycorrhize seedlings of these Scots pine with an EMF partner. In this case, this is the Piloderma olivaceum that we saw earlier in the presentation. And basically, when it's mycorrhized, it has better survival in the field. And so Louis is attempting to move this project forward, and I think he's doing a good job. And you can see here that instead of agar media, there's a more textured media here, which is like vermiculite and maybe some peat moss, and that's hydrated with the nutrient solution, autoclaved, and then put into these petri dishes. And then you have you inoculate the fungus into that petri dish, and the fungus grows throughout the media, and then you put a germinated seedling in through a hole in the top of the petri dish, and it grows its roots down in there into the media and becomes mycorrhized. Um, and I want to say, there's plastic. I talked about not using plastic in a couple slides ago, and I'm realizing, this looks like there's a lot of plastic use here, and there is some plastic use, but this is not at the same scale as saprotrophic mushroom cultivation, because eventually, these seedlings will be outplanted into the field, and then the bulk fruiting substrate will just be the soil. And when I gave this presentation once before, I had a comment about that, so I just wanted to note that. Um, okay, so once you have a mycorrhized seedling, so we kind of touched on this first box here. We have mycorrhizal synthesis happening in a lab, which can take 3 or 4 months or so. Well, there's still some steps that need to happen before you have mushrooms growing out of the ground. And so the next step is to basically move these mycorrhized seedlings into a non-sterile but a clean nursery environment. And these seedlings are up potted into non-sterile pots and some soil media. And this allows for the plant host to grow taller, but also increase their root biomass. And you're hoping that the fungus also continues to colonize the new root biomass. And so this is called acclimation. And I think it's called that because you're acclimating to like a non-sterile lab, or you're acclimating from a non-sterile environment to a nursery, or I'm sorry, you're acclimating from a sterile environment to a non-sterile environment. And so after acclimation, if it's successful, those seedlings can be outplanted into the field. And it's good to basically monitor that the ectomycorrhizal fungus is persisting on the roots. And as soon as 3 years after outplanting, there can be an onset of fruiting from the ectomycorrhizae. And this is an example here of saffron milk caps that are partnered with Monterey pine in New Zealand, and they are fruiting there. And then, of course, as scientists, we see a fifth step of this process here is to collect yield data, which is important for, I would say, attracting future investment into this field of research, because you have to know, how much bang are you getting for your buck here on these kinds of systems. Now, another way to establish a mycorrhizal synthesis between an EMF and its host is from spores. And this is primarily done for ascomycete EMF. So this would be in the tuber genus, the truffles. And basically these truffles are just like a spore mass. And so you have the spores produced on the inside of the truffle, and you can just take these and grind them up in some water and make a spore slurry. And so this seems pretty simple, right? It's like, wow, that's great. I could do that in my kitchen. And this spore slurry can be used to water seedlings of a suitable host or put into a planting hole. And so this is the commercially viable practice that's used for tuber borkii, the bianchetto truffle, which is a European species of truffle that's widely cultivated in Europe. And these truffles fetch $350 a pound. This can also be used to mycorrhize seedlings with tuber aestivum, which is the summer black truffle. And this is another European truffle, and this fetches about $380 a pound. This can also be done for tuber melanosporum, the black paragourd truffle, which fetches a modest $960 a pound. However, this doesn't work for the big kahuna, the tuber magnatum, the Italian white truffle, which is supposedly the most exquisite taste experience that you'll ever have. I've never had it, so I can't attest to it, but it costs $5,600 a pound. And unfortunately, this technique just doesn't work for this fungus. I'm going to touch on later, I tried something kind of similar to this using chanterelle and hedgehog mushrooms on hazelnut, and we'll talk about that later at the end of the presentation. And yeah, spoiler alert, it didn't work. But I'll walk you through the process. And I think it may be too early to tell. Perhaps it will still work. We'll see. Okay, so now that we've reviewed some of the methods that have been developed for cultivating these fungi, I want to jump into the case studies. So we have chanterelles in Sweden and Japan. We'll start there. So, the chanterelle story is long and winding, and for how long it's been researched and been ongoing, there hasn't been a lot of gains until recently. And so let's start at the beginning here. In 1979, the first pure culture of Cantharellus cibarius was obtained in Sweden. And in 1989, the first mycorrhizal synthesis was obtained with Cantharellus cibarius on Scots pine by the same group of Swedish researchers. And in 1996, a different group of Swedish researchers obtained the first fruitification event, artificial fruitification event of chanterelle in a greenhouse setting. And you can see that is the mushroom there, the famous mushroom. And what you can't tell from the picture is that it's actually protruding from a hole and a drainage hole in the bottom of its nursery pot. And so it's a pretty haphazard occurrence, and clearly this technique hadn't been mastered yet. In 1997, this group of Swedish researchers founded a company, Cantharellus AB, and this company was established to commercialize Cantharellus cibarius mycorrhized seedlings, specifically Scots pine for planting out in Scandinavia. However, after a few years of research, basically the techniques that were being used, the cost of those techniques were not being justified by the potential revenue of selling these seedlings, and so investors pulled their money, and this company shuttered its doors, unfortunately. And this research could have ended there, and it did for a while. However, in 2019, a paper came out released by a Japanese research group that published a protocol for the isolation of Cantharellus anzutaki, ectomycorrhizal root tips. And Cantharellus anzutaki is a sister species to Cantharellus cibarius, and it looks just like the golden chanterelle. And this was a big deal because getting an isolate of an individual chanterelle mushroom was extremely difficult, and the odds of success were super low. So isolating from fruit body tissue was really difficult. Isolating from spores was very difficult. However, this group went out and sampled ectomycorrhizal root tips, sequenced those root tips to confirm the identity of the fungus, and then isolated, basically meaning putting those root tip tissue onto synthetic media like petri dish media and getting the fungus to grow out onto the media successfully. And this worked basically orders of magnitude better than isolating from fruit body or spores. And so now there's basically they had a large collection of isolates to work with, which is important because there's a lot of variation between individuals. And so with this large collection of isolates, they inoculated red pine seedlings and Conara oak seedlings in the greenhouse. And from these inoculated seedlings, the seedlings were mycorrhized, and then they observed 29 fruitification events over the period of 2 years from these mycorrhized seedlings, which is kind of astounding. And some of the important findings from this study included, what's pretty intuitive is that larger jars produced larger mushrooms. And so you have more host biomass supporting more fungal biomass, which supports larger fruiting bodies. Less intuitive is that mycorrhized seedlings grown in organic soil produced no fruiting bodies. However, so all 29 fruitifications occurred from mycorrhized seedlings grown in mineral soil. And you can see the stark contrast in host health between the red pine seedling growing in mineral soil and that growing in organic soil. Clearly, the mineral soil is not conducive to a really healthy seedling. And so it looks like it's nutrient deficient and it's stunted. And so does this mean that future growers of this kind of system need to stress out their host trees? I don't know. It's too early to tell, but it's an interesting finding nonetheless. And lastly, from this paper, the researchers um, uh, found that there was a significant interaction between the individual genotype of the fungal isolate and the individual genotype of the host. And so it's not enough to just match up the species of EMF with the species of host. You actually have to match up the correct individual or a good individual from the fungal population with a receptive individual of the host population. So you have to have a good match there. And so in 2020, these mycorrhizal seedlings were outplanted into the field. And as of 2022, I read a research update, and there are so far no yields yet, but it is pretty early. And the authors speculate that the fungus is still accumulating biomass before it might set fruit. So that's the chanterelle story. Now we'll move on to the saffron milk caps. And this is a story, I would say, of really great success. These researchers in New Zealand uh, basically in the early 2000s were able to mycorrhize Scots pine and Monterey pine seedlings with saffron milk cap successfully. And after they were able to repeat this and do this well, they outplanted these mycorrhized pine seedlings into orchards in 2008. And then they collected mushrooms from underneath individual trees over 11 years. And the first mushrooms appeared in 2010 underneath the Monterey pine and in 2012 underneath the Scots pine. And so these mycorrhized seedlings were planted out in separate plantations. And the mushrooms first appeared in the Monterey pine plantation, just 2 years after outplanting. And so this is a look at the yield data for each of these pine plantations. And so on the y-axis, we have the yields of saffron milkcap mushroom in kilograms per hectare. And then on the x-axis, we have the year that those mushrooms were harvested. So we have orchard establishment in 2008. The first mushrooms appear in 2010 in the Monterey pine, Pinus radiata orchard, which is the blue line, and they appear 2 years later underneath the Scots pine trees. And you can see for the Monterey pine, production peaks just 4 years after they began appearing in the plantation, and then reduces to a modest 200 kilograms per hectare. However, for the Scots pine, even though they're slower to get going, they peaked about 5 years after they first appeared. However, they haven't reduced to a non-commercially viable amount of harvest. They've stayed pretty high, above 700 kilograms per hectare. And you can see the difference in these pine plantations. This is photographs that were taken of each plantation in 2019, and the Monterey pine trees grew much faster than the Scots pine. And basically the canopy became closed. And what the authors noticed was that the canopy closed in 2014, which is when the production of mushrooms steeply dropped. And so they speculated that it's something to do with canopy closure that reduces yields. And you can see in the Scots pine, there's still a lot of edge between each Scots pine tree, and there's a lot of sunlight still hitting the floor of the plantation. Now, just to further tease apart this hypothesis, so the authors, again, you have to remember that they sampled mushrooms from individual trees, and so they kept track of which mushrooms were appearing by which trees, and they separated the trees in the Monterey pine plantation into trees that were located at the edge of the plantation and those that were located at the inside. And so the edge trees are represented by the black line, and the interior trees by the orange line, and the cumulative number of mushrooms harvested from each category of tree is on the y-axis. And so we have similar amounts of mushrooms being produced by interior and exterior trees until 2014, which is when the canopy closed, and then you have a divergence, and you have interior trees producing less mushrooms. And so there's something to do with this canopy openness, or perhaps an edge effect occurring here. And so this is interesting with thinking about agroforestry systems that might be suitable for an ectomycorrhizal fungal component. These systems need to have lots of, I guess, canopy openness, at least in this this kind of system that was studied with the saffron milkcap. Okay, and now we'll bring it back home to the pecan truffle double cropping as it's been explored in the southern United States. So the pecan truffle is a lesser known species of truffle. This is native to North America. It has a very broad host range despite its name. So it grows both with pecan trees, but also with red and white oak, and also hazelnut. And its geographical range spans from northern Mexico all the way up to Ontario, east of the Rocky Mountains. This fungus will form fruiting bodies from mid-September through December. And these fruiting bodies go for more than $1,200 a pound. There's only one company that I could find that sells them, the Truffle Dog Company here in the United States. And they're currently out of stock, but I saw that back when they were in stock, they were $1,200 a pound. So these are these are precious mushrooms. And so there's been a lot of interest in their potential cultivation and as a double crop with pecan. And so pecan management and pecan plantations lends itself well to the creation and maintenance of suitable habitat for the pecan truffle, namely the removal of pecan leaf litter, as well as high nutrient inputs, so having a lot of nutrients in the soil, mowing and herbicide to reduce herbaceous plant cover on the ground, and liming the soil to increase pH. And tuber species are largely disturbance adapted fungi, and so they like it when you remove the litter and when you have high nutrients and when there's a high pH. And so these conditions are suitable for the fungus. And so how do I go here? This group of researchers here decided to start exploring the idea of double cropping the pecan truffle with pecan trees. And what they did in their first study is instead of doing an empirical study and inoculating pecan seedlings with the pecan truffle, they simply did an observational study to see if does the pecan truffle does it exist in symbiosis with pecan trees in pecan orchards just naturally, just with natural inoculant levels. And then in the pecan orchards that they're in, what are the conditions that seem to determine their presence in an orchard? And so how they went about this research was they sampled 25 orchards in Georgia and Florida, and they sampled 3 trees per orchard, and all these orchards had a diversity of soil types. And they also sampled 1 forest tree in the natural forest adjacent to the orchard at 15 of these sites to get an idea of the comparable, does the pecan truffle also exist in the natural forest area? And they also conducted soil nutrient and pH analyses to try and understand the conditions that lead to the presence of the pecan truffle in these orchards. And they performed high throughput sequencing of over 9,000 sampled ectomycorrhizae. So it's a lot of work. And they had sequence reads from this extracted DNA that matched 96 different species of ectomycorrhizal fungi. And so this is not only a study to look at the presence or absence of the pecan truffle, but also looking at the whole community of ectomycorrhizal fungi that exists in these pecan orchards. And this is a graph that shows some of the results. And basically what you're seeing here on the y-axis, above 0, we have the frequency that a fungus was found. And so on the x-axis down here, we have the different species of fungi that were found in the study. And then below the 0, we have the number of sequence reads. So basically meaning how much DNA of this fungus was found in the sample. And so this indicates um, I guess the abundance of the fungus on a particular root tip sample versus above is how many trees that fungus was found on. And so if we look at tuber lyonii was found on 22 of 76 pecan trees that were sampled, which is the 6th most abundant fungus on a per tree basis, but it was actually the most abundant fungus on just a fungal biomass basis. And that's, you can see that here. And the black shading indicates samples from pecan trees in the pecan orchard, and the white shading indicates just a natural forest tree in an adjacent area. And so you can see that pecan truffles were both present in an adjacent forest area as well as in the plantation area. And this was a trend that was mostly true for a lot of these species of fungi that were found in the study. And in fact, there wasn't a significant difference between the fungi found in natural forest areas and those found in pecan orchards. And so the upshot of this is that likely there will be movement of fungi between the natural forest area and a pecan plantation. And so this is something to consider when you want to really drive the fungal community in a pecan plantation to be exclusively tuber lyonii, because you might have other species of fungi coming in. And there's actually 7 other species of tuber in the truffle genus that are undescribed, so they're not formal species, but different operational taxonomic units that were found in the study. And these undescribed tuber species are likely the most fierce competitors with tuber lyonii in a double cropping system. Okay, the authors also found a dramatic effect of soil pH on the ectomycorrhizal fungal community composition in these orchards. And so what you can see here on the x-axis, we have the pH at the sampled orchard. And then on the y-axis, we have the relative abundance of different lineages of fungi at those levels of pH. And I want to point your attention to the 2 Ascomycota lineages here, Galactinia and Tuber halvella. These 2 lineages really increased dramatically with increasing soil pH. And so the soil pH is a strong filter for the community, fungal community composition, and tuber really benefits from this. And so again, the liming of the soil in these pecan orchards is advantageous if you're wanting to grow tuber species. Okay, the second study I'm going to cover here in this line of research looks at, so what is the kind of a more empirical study that looks at artificial inoculation of pecan seedlings in a pecan orchard with the pecan truffle. And so this addresses the question of, can the mycorrhization of pecan seedlings with the pecan truffle be integrated into the standard pecan nursery protocol? And so this is kind of looking at would this be really economically feasible? Is this something that could be done at a massive scale and integrated into the current system of growing pecans? And so to understand the study, first, I want to give a little bit of background information on raising pecan seedlings. And so first, how this is done is that these are all grown in outdoor beds, and the soil is tilled and fumigated. And you can see a picture of that happening here with the plastic, which functions both as weed control, but also for fumigation to kill any kind of microbial inhabitants in the soil. And then seeds are planted into little holes that are punched into that plastic. And then after a year of growth, those seedlings, the rootstock is grafted with scion wood of a preferred cultivar of pecan. And then after 2 years of growth, those seedlings are extracted out of those nursery beds and transplanted as saplings into a pecan orchard. And there we are. We have the 2-year-old seedlings there ready to go. And so what the authors did is that they simply put a spore slurry of the pecan truffle into the planting holes, right as the seeds were being placed into those holes. And in this experiment, they varied the inoculum concentration. So in the inoculum, is really expensive stuff because, these truffles cost a lot. And so it's an important question to know, how much inoculum do you need to have successful mycorrhization of the pecan tree? And so they varied inoculum concentration, and they did this at 3 sites. And they sampled the root tips of these saplings after 2 years of growth. And they performed high throughput sequencing of the ectomycorrhizae. So basically sequencing the DNA of the mycorrhizae that they found on those root tips to verify the identity of those fungi. And this is some of the results here. This basically shows the effect of inoculum concentration on the success of tuber Lyoneae colonization. And so on the y-axis, we have the percentage of total reeds of ectomycorrhizal fungi. And so they've basically found other species of fungi on these root tips too. And species that didn't belong to tuber Lyoneae are shaded in this black here. And species that belong to tuber Lyoneae are in the diagonal shaded lines. And this column here will add up to 100%. So it's just a split. What belongs to tuber Lyoneae and what belongs to other fungi, what you could say are weed fungi in this case. And so on this x-axis, we have basically 2 grams of inoculum concentration, and then decreasing concentration as you move across the axis, all the way down to 0.02 grams. And you can see that at as little as 0.1 grams of spore slurry, you have dominance of tuber Lyoneae on the root tips of those saplings. And so that's an encouraging result. And this paper came out in, I think this one was like early 2020s, and I haven't seen anything since. And so I'm curious as to where, maybe the research grants lapsed funding or where this is at now, but this appears to be pretty good results. Okay, and so lastly, I just want to quick touch on an experiment I did because I thought, hey, if how do these fungi do it in nature? How do they colonize new hosts? And you would think that because these fruiting bodies, mushrooms, these EMF fungi produce mushrooms not to necessarily for the purpose of feeding animals, but for the purpose of spore dispersal. And you'd think that those spores serve a function, namely to disseminate the fungus to new hosts and colonize new hosts. And so I thought, can spores be used as an inoculum? And can it be done low tech? Does this have to be done in a really sterile laboratory, or can this be done low tech? And so what I did was I went out and collected chanterelle mushrooms, as well as 2 species of hydnum. And we have hydnum aerostatosporum and hydnum repandum, and collected spore deposits on aluminum foil. And you can see the hydnum aerostatosporum basidiospores here. There were a whole bunch, like, what was it, like maybe 100,000 per mil of inoculum. And then I also got some hybrid hazelnut seedlings from a friend of mine here at the university who is working to grow hybrid hazelnuts in the Midwest. And these hybrid hazelnuts I thought could be a suitable host because hazelnuts are known to associate with a wide diversity of ectomycorrhizal fungi, including specifically hydnum aerostatosporum, which was reported by a paper that Peter Kennedy authored a number of years ago. Also, it's been reported as a host for chanterelle. And so I got these seeds and I basically surface sterilize them in a weak bleach solution by just soaking them in some bleach to kill any kind of fungal spores that might be on them. And then I rinse them in just tap water to rinse off the bleach. And then I planted them into these flats in a vermiculite, so just a mineral soil that likely doesn't have a lot of spore load in it. And sure enough, it had decent germination results, maybe 30 to 40% of the seeds germinated. And so I took my basidiospores, and after the seeds had germinated and there was a small shoot, and then just put like, what was it, maybe 5 mils of basidiospore solution along. There was also some nutrients in there. Special media for ectomycorrhizal fungi. And so the spores had the nutrients, they had the moisture, and now they're in contact with a host plant. And so I monitored these seedlings for 3 months, and then I pulled them up by the roots. And this is not a photo of mine. I didn't actually take a photo, but this is a hazelnut seedling, and with its roots exposed here. And what you can see is that the hazelnuts are really a hairy plant. And you can see the hairs on the stem here. And those hairs also are equally prevalent on the roots. And so I thought at first, like, oh my gosh, look at all of my mycelium. This is awesome. But I was skeptical because I've seen Cantharellus ectomycorrhizae appear yellowish in color. And so I thought, why is it not pigmented? And sure enough, I pulled up my control hazelnuts that I had not inoculated with anything, and the roots looked exactly the same. So, kind of anticlimactic. But it's also possible that the spores just haven't germinated yet. I read that it has taken, in some instances, Cantharellus spores 8 to 10 months to germinate. And so I've kept the seedlings. They're in the greenhouse, and we're going to plant them out at my advisor Bob Blanchett's house this summer. And maybe someday you'll have mushrooms growing. But I think, this mycorrhizing seedlings wasn't as easy as I thought it could be. And that's kind of one moral of the story. But also, I think that, as I'll get to here in my last summary slide, this is really a new field of research. There's so many things that could be tried. And although there's lots of challenges, there's a lot of opportunity. And so to summarize, pure cultures continue to kind of be a challenge. These fungi don't grow that well on artificial media. A lot of them don't. And even just obtaining an isolate can be difficult. Performing the mycorrhizal synthesis is pretty tedious, which is why I was amenable to the idea of using spores. As you can see here, this is carbon filter paper in a petri dish, and these are birch seedlings that are being mycorrhized with a fungus. And doing this for thousands and thousands of seedlings to be outplanted just isn't really practical. And so the costs of doing this process are just kind of outweigh the potential revenue as it stands. Also, field experiments evaluating the cost benefit of these systems are decades long. And because they're decades long, it's hard to get funding to maintain those experiments. And it's kind of a catch-22 because if you don't have the experiments, if you don't have the funding to do the experiment, well, then you don't have the results to attract investment. And so this has left us with a dearth of field studies with yield data. But like I said, this is a nascent field. This is, I think, rife with opportunity, and there's just so many things that could be tried. And I hope people try them, and I think they will. I want to thank Professor here at the U of M, Peter Kennedy, as well as Bob Blanchett for helping me think about this presentation and what I might want to include and providing me feedback. And also my fellow graduate students here in the Department of Plant Pathology at the university for providing me some feedback on this presentation. I had a lot of beautiful photos in the presentation, and a lot of them weren't mine. And so I just want to credit the people who took those photos on the slide here, and then some sources for the text that I had in the presentation. Yeah, and that's it. I know I kind of went maybe a bit long, but I'm happy to answer any kind of questions you guys got.
It was wonderful, Ryan. Thank you so much for that. We do have a few questions that have come in. One is from Peter Martinaco, and he asks, what percentage of the inoculated pecan seeds were successful? Do you happen to know that?
Oh man, what percentage were successful? Well, I don't know if they calculated that metric. I know that
Okay.
so the results that were in the paper were basically of all the saplings that they sampled, it was like, gosh, let me just pull up my slide there. It was like I think it was like greater than 80% of the ectomycorrhizae on those root tips belong to tuberlionii. And I would kind of
Nice.
make, I don't know, I feel like a pretty safe assumption that a lot of those saplings did have the fungus.
Mm-hmm.
Um, it's a tried and true method of inoculating using tuber species,
Mm-hmm.
they're well adapted for it. And I would think that most of those saplings were colonized.
So my own question off of that is, does does having those ectomycorrhizal the tubers on there, does that benefit them, to make them more successful, or is it a wash?
Yeah, so does it benefit the pecan tree?
Yeah.
Yeah, I think that is something that would have to be explored. And I didn't see that being explored in what I read. But there can even be a difference. A lot of time um, these symbioses, the benefit to the host plant is context dependent. And so maybe
Mhm.
there's a huge benefit in drought conditions, but in adequate rainfall conditions, there's actually a slight decrease in host fitness. But
That
what I
makes
would
sense.
think is that, these symbioses are so prevalent in nature,
Mhm.
and these lineages of fungi are so successful,
Mhm.
and there's so few plants that don't have these kinds of relationships, is that as a
Mhm.
whole, that probably leans towards being beneficial. But it could even depend on the individual genotype of fungus being used to inoculate. Does it tend towards cheating, or is it
Mhm.
really true in its exchange?
Okay, nice. Thank you. Aaron asks, do you have a thermometer, or PCR the old-fashioned way? He's in the planning stages of, so he's thinking about, should he just get a lab's recycled thermometer if that was part of the plan?
Yeah. So you want to basically do like fungal DNA extraction and sequencing, and you're wondering if you can do, um, yeah. So yeah, what's referred to as endpoint PCR is like I
Mhm.
think what a lot of people think about is like being the classic PCR approach. And yeah, we do that all the time in our lab.
Mhm.
That's pretty much what we use. High throughput sequencing is nice because you don't need to culture the fungus.
Mhm.
You can extract from, gosh, just a block of soil, and see what's in there.
Mhm.
And so you can get a lot of data really fast. Um, but if you want to work with a particular fungus, I think it does make more sense to do endpoint PCR. And so, yeah, getting a recycled thermocycler or a recycled thermocycler sounds like a great idea to me.
So my personal question then, is, how do you deal with contamination issues? Or is that not such a big deal since you're working out in the open?
Yeah, so yeah, that's a good question. So contamination, I guess, can occur at like any of these steps of the cultivation process. It can occur when you're growing your fungus on a petri dish and you have a fungal spore in the atmosphere in your lab or at your home just lands on the dish and takes off really fast. Um, and a lot of these like trichoderma or cladosporium or alternaria are just really aggressive and fast growing. And so really if you want to do the mycorrhizal synthesis using mycelium and not from spores, you really need a lab space to do that because otherwise you'll just get weed fungi growing on your on your seedlings.
Mm-hmm.
But that's why the spore inoculum is so attractive from a scaling point of view because you can do it. You could do it in a pole barn. You could do it out in the field because these spores are very aggressive and they're adapted to those kinds of
Mm-hmm.
soil environments. So yeah, it depends on what you want to work with. If you want to do truffles, yeah, I think you could totally research this without a lab space.
Okay, nice. Elena asks, how old are your hazelnut saplings?
They are, let's see, now I planted them in September, so about 5 months.
Okay, nice.
Yeah.
And somebody else says, PC says, mind blown. I have an amazing spot for chanterelles, and there are so many hazelnuts there. I didn't know that that was a thing.
Yep. Yeah. Yeah. And yeah, I mean the you might even have, chanterelles coming up naturally in a hazelnut area. Um, and I guess jumping back to the contamination bit, something else I wanted to mention was you can't really inoculate a hazelnut shrub or an oak tree or something that's already growing in the field because it's probably I'd say almost certainly already colonized by some native ectomycorrhizal fungi. And so that's the other contamination piece is like you can get contamination. You've had successful mycorrhization in the lab and you outplant your seedlings, but then something else comes in that's more competitive and displaces, your target fungus. Um, and so, yeah, do these relationships persist over decades or however long you need to recoup your investment? And so, yeah, that's another question that's kind of, still unanswered for a lot of these systems.
This might seem like a weird question, but I was thinking about this as you were showing us that image of the different forests and where the ectomycorrhizal growth is most prevalent. So, do we know what tree? Is there a single tree that is like the thing? The most. I mean, I think of when I go out, I love looking for oak trees because of the different mushrooms that I find in conjunction with that. But are there particular trees that are most prevalent, or do we find ectomycorrhizal mushrooms being kind of just spread out fairly evenly throughout a variety of trees in those areas?
Yeah, that's a really good question. I think it's probably pretty nuanced. Like different trees will associate with different EMF species, and some trees are known to associate with a really wide variety. Like hazelnuts will kind of the study that Peter Kennedy did, where he found the hedgehog mushroom with hazelnut. That same study, he basically looked at the EMF community in a pine-dominated stand and then an oak-dominated stand, and the hazelnuts as a shrub layer in both. And what he found was that the communities were quite different in the oak between the oak and the pine stand, but those associated with the hazelnut were kind of the hazelnut was kind of like a bridge between those communities, and the hazelnut is associated with fungi from each community. Um, and so, yeah, I guess another kind of way to answer your question is like, okay, what about like which tree is the easiest to mycorrhize with a fungus? And I was talking with my friend Louis, who's the guy studying in Scotland.
Mhm.
He said, man, instead of trying to do hazelnut and hedgehog, you should just do Scots pine and saffron milkcap. And as you
Mm.
saw from the presentation, that was really
Mhm.
successful. And I
Yeah.
guess that relationship is just like easy to synthesize because they're
Mhm.
both I guess, aggressively colonize one another. Um,
Nice. So
so
like a really strong partnership.
yeah, yeah. It doesn't take a lot of convincing.
Mm-hmm. Cool.
Olena, I think you already unmuted yourself, right? It looks like Olena is unmuted for her question. If you can hear me. And I was also going to invite John to unmute because you had a couple early on questions and
Yes.
you had this
Yeah.
here to just ask.
Well, I just had one. There's been so much talk about morels, and are they EMF or are they saprobes? Does science have a stance yet?
My understanding is that they have the capability to do both. And so it's one of those things where like we try to put organisms into these neat categorical bins, but the lines of those bins are really quite blurry. And so it depends on the context. Like if their host tree is alive and giving them a lot of sugars, then they might not have a reason to engage in saprotrophy. But if their host tree dies or there's some other kind of circumstance that may push them into, turning on those genes to engage in saprotrophy. Um, yeah, I'm a morel expert, but these are being cultivated in China en masse, and how they do this is they basically grow them in on these sawdust blocks as a saprotroph because morels will grow really well in culture. You can get them to grow on a plate easily. You can get them to grow on a sawdust block really easily. But for a long time, people didn't understand how to trigger fruiting
Mm.
until scientists in China basically grow them in these sawdust blocks and put the blocks into a bed of soil with some nutrients in it, just like in the greenhouse. And then the fungus will basically colonize that nutrified soil as like a big, in a big row of soil, just like a garden bed or something. And then they won't produce mushrooms until you put a non-nutritive bag of, or is it nutritive? No, I can't remember now if it's nutritive or non-nutritive. I think it's nutritive. Another nutritive bag of sawdust, a block of sawdust on top of the bed with the bottom end open. So the fungus grows up and colonizes that little pocket of nutrients, and then for some reason that initiates primordia development. And so whoever thought of that, is a creative person.
Mhm.
Their
Wow.
success, though, was primarily with a strain that's known to be primarily saprobic. It's like, I mean, you're not going to get that with our state mushroom because they haven't unlocked that key yet.
Yeah.
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 Yes. This is completely anecdotal and it's probably just, I mean, we're always seeing things, right? But whenever I'm washing mushrooms, I always dump my spore water out in my yard on something. Do you guys do that too?
Mhm.
And this was one of the very first year. And I remember I went to look for morels with MMS. And of course, Lee did his usual, who's never found a morel? And points went out to me. And I brought my stuff home and trimmed them and dumped them out. I'm telling you, it was 1 year later in my yard. There also happened to be a dead elm, but there were morels in my yard the next year. They might have always been there and I just didn't have the eyes for them. Or maybe because I dumped new spores there, or maybe because that tree just died. I don't know what it was, but it felt magical. And I was like, I planted.
That's so sweet. You probably did plant seeds. You just happen to have a nice place to
The
plant
spot
them
for him. Yeah,
because of
actually,
the dead elm.
I asked Tom Volk, and he said sometimes you could have things growing mycorrhizally that aren't fruiting. And if you've introduced different spores, you sort of introduced either another mating um, type or another mating organism or a competitive organism that you could have pushed it to do something, which I thought was fascinating as well.
Nice. Olena, have you figured out, can you talk now? Why don't you type it in? Charles Kilmer says, I have dumped so much more morel water around my yard and no luck, and I keep hoping, though. We all do. We all keep hoping.
Ryan, I was going to mention one more thing, and I was when you guys were talking about this symbiotic nature or the mutualistic nature of the mushrooms and the plants, and if they're always helping the plants. And I also just saw a lecture by Christian Schwartz, and it was so fascinating. And he was citing Suzanne Simard's work and said, because you're studying something in a lab, it's a lab, right? So you've removed the exterior influences. And when you're studying something in nature, especially if it's mycorrhizal, once you disturb it, you've disturbed it. So you don't know what would have happened. But what they had kind of found was, I don't remember which study it was, but the mycorrhizal nature of a certain mushroom that was in a forest, it was like it affected the trees differently based on the age of the tree, the water of that year. You know what I mean? It was sort of like this fluid thing. And they were saying for some of the younger plants, the mushrooms maybe weren't really helping it. It was maybe taking too much. But then when the plants were strong, the plants were giving back to the mushrooms. And it was this back and forth where some years it helped the mushrooms more, and some years it helped the plants more. Mm-hmm. And it's kind of you can't really test that in a lab because you've removed all those external things. And I thought that was kind of a fascinating way to look at it, that we want to have a word that says this is mutualistic, or this is the mother tree, or this is, you know what I mean? We attach all of these kind of feelings to these words, and in reality it's changing all the time, and it's a hard thing to quantify and measure in science.
Like any relationship, really.
Yeah, right. Yeah, absolutely.
So
Yeah,
PC
cool.
asks, were there hundreds of saffron milk caps per tree in those groves?
I would think, I don't know about multiple hundreds, but I would think approaching 100. I mean, 1,100 kilograms per hectare. That's a lot.
Mm-hmm. Well, and if you walked around any tree in any forest and you found even close to 100 mushrooms around it, you'd be pretty excited.
Yeah. Uh-huh.
Oh, that's cool. Alina, were you able to ever, are you still here? Well, I know your email address. So if she gets her question to me, I will pass it on to you. We thank you so much for your time tonight. Every time that you come and you talk to us, I learn something new, and I really enjoyed this one. So thank you so very much.
Yeah, thanks for having me. I just thought of a question for you all.
Yeah.
I
Uh-huh.
was curious about, so this idea of fruitification happening on the edge of a mycelial mat. And do
Mm-hmm.
you all feel like you see that when you're out foraging, like at the
Yes.
edge of an oak stand or something, you see more fruitification occurring than on the interior?
I know that happens with wine
Mm-hmm.
caps.
Bullets as well.
Yeah. Heather, do you hear me now?
Yes.
Finally, I have a question. Sorry, I apologize. I ended up dropping through the computer and join the iPad. Ryan, thank you very much. Very interesting presentation. Really much appreciated you doing it. But have you ever thought of those friends and foes? As you can say, it's a contamination for a certain act. But I will feel like, in my personal experience, when I try to do, let's say, rough shot, not the petri dish seeding of the chanterelles, is like you need to seed some additional mushrooms, that is, can inoculate the tree roots prior to the chanterelle setting the place to grow. Like, like, let's say, certain russulas, certain amanitas that grow with a specific type of chanterelles. Let's say, because let's say Peter can attest, I have a spot that I've been seeding roughshod quite a bit, and it's been producing consistently for the last several years. And it's nothing around it. You can look for mushrooms within like half a mile and you cannot find them. But in that particular spot, you can find chanterelles all the time.
Okay. So you've been dumping chanterelle spores there?
Not the spores, but chanterelle soft cuts. Let's say I'll find the chanterelles, whatever half spoiled, whatever I have cleaning in there, and I'll dump it in a specific spot. And it grows. It grows like crazy. Like last year, we had like half a basket, and there we just went in. It's like, and absolutely nothing anywhere else.
Yeah, I would say try doing that same thing in another spot and see if it works.
I
And
mean,
if it does, let me know.
I mean, the thing is, it's like it is at several different spots, and I have my boletes growing in there, and I have like tricholomas, which is like Tricholoma equestria. You can spore them, and I had my boletes coming out.
It's awesome.
But
yeah, cool.
not hedgehogs. I'm trying this year is like I'm having the black trumpets, but it's like I've been kind of experimenting in a lot of different things.
Yeah, cool.
Yeah.
Thanks for sharing.
You're welcome. But it is, you can grow chanterelles. I can guarantee.
Yeah. I think, I mean, it's hard to know, like I said, like repeatability is really important. So if you can repeat what you did with the chanterelles and a few other spots and then write down exactly what you did, then maybe other people could try it too.
I will say it's within 3 to 5 years. That's unfortunately thing is like it's timing. You need to wait that long. It's not like like if you grow like white like button mushrooms, you can get the results, or any aggressive mushrooms within 1 year. Anything whatever is like progressive, you need to have kind of semi-prepared grounds in order to have it. And it's you need to have a mix, because it is like chanterelles does not grow alone. They grow in kind of like friend combination with other mushrooms. Therefore, it's like you might be something like you plant chanterelles and you plant amanitas and russulas together. Have you ever tried something like that?
I've never tried. So you're basically like taking some fruiting bodies and then bringing them to a natural forest area and then putting them there? Or are you putting them like over the top of seed saplings that you're planting or?
In places in the forest.
Yeah, I don't know. That sounds cool. I would think that there'd be already fungi that would already be there that would be inhabiting the roots of those trees that would make it hard for them to establish, but I don't know. I mean, these systems aren't really well studied, and if you're seeing something happen on a repeated basis, and that's cool.
I mean, my question is going to be like Sualis needs to grow on a young Sualis, like slippery jacks, needs to grow on a young pine, which is in further will be conducive to growing boletes. If you didn't have Sualis, no boletes will grow. You kind of need to have something already grown on the roots in order to have next mushroom, next generation grow.
Yeah, so some kind of like succession.
Yes.
So what did you say? The Psilocybe comes first and then the boletes?
Yes.
Yeah, that's cool. I don't know. I don't know about that, but that sounds neat.
Because Sualis grow within like 5 years to 10 years, and boletes grow from 10 years forward.
Cool.
That's it. Heather, back to you. Thank you.
Thank you so much, Ryan, for all of your time tonight. And we want to be respectful of your time and let you go. But we greatly appreciate your knowledge and your generosity of sharing it with us.
Yeah. Thanks for having me, everybody. It's always fun to come and visit. come and
Thank
talk.
you
I'm
so
so
much,
busy
Ryan.
with a 3-year-old, so I don't get to participate in a lot of other ways, but this is a way I can be here. So
Thank you so much, Ryan. Really appreciate it.
Mhm.
yeah,
Yeah.
you're welcome, Peter.
This was awesome. Thank you.
Yeah, you're
Thank
welcome.
you. Bye now.
Bye.